So, hello students, welcome to Vedantu NEET English. This is Basavaraj Sir, your biology master teacher. Students, good morning each and every one of you. So students, quickly let me know in the chat right now if I'm audible and visible to each and every one of you. Quickly let me know in the chat. If I'm audible and visible, amazing. Okay. Now students, this is going to be one short lecture on the plant growth and regulators. After this, the entire plant physiology is over one more time. We finished the respiration plants as well as photosynthesis and higher plants, both are done and dusted. Okay. So students, quickly let me know in the chat right now if I am audible and visible to each and every one of you. Quickly let me know in the chat, students. Yes. Now with that being said, let me tell you right now honestly, the weightage of this chapter. Now this chapter generally 4 questions. Generally 4 questions and 16 marks you'll get from these chapters. Now all of you are aware, right? There is no more section B, students, no more section B, and you have to attend every single question without any exception. Now this is a chapter which will help you in that. How is that? Because this is a chapter where we can tell you exactly from where the question will come and exactly what is the difficulty level. Because in today's class, I will teach you the concept entirely, but I will also show you the questions which you can expect from this chapter. So with that being said, students, can we start today's class? Quickly some fire emoji in the chat, some green hearts in the chat to welcome today's class or to start today's class. Okay. Now, so obviously it's going to be one shot, right? One shot. So we'll be learning about growth, the entire parameters of growth, measurement of growth, entire concept. And let me tell you, if you have zero knowledge, absolutely zero knowledge, this is a class for you. Because I'll be going from all the way from the basics to the advanced level, all the way. Okay. Then we'll be learning of differentiation, dedifferentiation, redifferentiation. Then the development part. And this right here, student, this right here, plant growth and regulators. This is the part where majority of the questions will be coming from. So I want everyone to be very active in this time. And let me show you, student, let me show you. For this particular part, right, this particular part, I have an amazing formula sheet for you. When I say formula sheet, I mean a particular list of every single growth promoter and their functions side by side. Do you see that? Cytokinins, ethylene, abscisic acid. So majority of your questions will be from these 3 slides, which we'll be doing in the summary later on. Okay. And let me tell you, students, before I start the class, this entire session PDF will be available to you in the description of this today's video after the class is over. Yes. So can we start, students? Can we start today's class now? Can we start? Okay. Let's open up the class with a question. As usual, every single class of mine, if you notice, I always open with a simple question. Now, all of you have finished the chapter Living World. All of you agree with me? Tell me, how many of you finished Living World? Raise your hands. So if you have finished the chapter Living World, there we learn something called as growth. Now, students, quickly tell me in the chat, is growth right here a fundamental feature or is it a defining feature? If you have learned the chapter Living World with me, you will tell me, sir, growth is a fundamental feature because there are certain exceptions here, Okay, fundamental feature. Now, at the same time, can any one of you remind me what is growth? I will tell you, growth is an irreversible change. But what are the twin characters of growth? That I want to know from all of you. Something called as twin characters of growth. That I want to know from all of you. What is called as twin characters of growth? Anyone, students? Every single growth will include these 2 things. Biological growth will include these 2 things. The first one is increase in mass and increase in number of cells. Number of cells or number of individuals. Number of cells or increase in number number. Now this right here is called as twin characters of growth. Now if all of you remember at the same time, there are two types of growth. How many of you remember? There are actually, growth can be broadly classified. Growth can be broadly classified into two separate categories. Okay. First one is called as intrinsic growth. Intrinsic growth. Second one is called as extrinsic growth. Extrinsic growth. Intrinsic and extrinsic growth. Now, what is the difference between intrinsic and extrinsic? Listen to me very carefully. Intrinsic growth is the growth from the inside. It is the growth from the inside. For example, me, you, all of us are growing from the inside to outside. Does anyone grow from outside to inside? Tell me in the chat. Does anyone grow from outside? No. All of us grow from inside to outside. Intrinsic. Now, what is extrinsic growth? Growth on the outside. Growth on the outside is extrinsic. Let me give a quick example for extrinsic growth. If any one of you visited Kashmir, during the months of November, December, January, there will be snowfall there. There will be snowfall. Now, during the time of the snowfall, if this is a mountain, the snow starts to accumulate on the top. All of you agree with me? The snow will start to accumulate on the top. Now, this type of growth, which is happening from the outside, can you see here? Growth which is happening from the outside is called as extrinsic growth. Extrinsic growth. All of you know. Now, let's get back to the famous definition. Growth is what, students? Growth is nothing but it is a irreversible permanent change. It is an irreversible permanent change. That is growth. Now I need one volunteer who will come forward and tell me in the chat. I need one volunteer now. Let me look at the chat and tell me. Let's pick up one volunteer now from the chat. Who wants to volunteer? Anyone? No, okay, students. Let me just check my phone. Why is the class not opening? Okay. I need one volunteer. Let's take. Okay. Let's take 𝐂𝐤 itself as a volunteer. Let's take 𝐂𝐤. Okay. Now let's assume that 𝐂𝐤 was, let's say, around 3 feet height. 𝐂𝐤 was, let's say, around 3 feet height in class 6th or let's say class 5th. 3 feet height in class 5th. 3 feet height. Now from 3 feet height, 𝐂𝐤 became, let's say, around 6 feet height when she came to class 12th. From 3 feet to 6 feet. Now students, tell me in the chat, is this growth? Do we call this as growth now? 3 feet to 6 feet height. Obviously it is a growth. Why? Because it is permanent and it is irreversible. It means that once you are 6 feet, there is no way again you can go back to 3 feet. It is not possible. So henceforth, I will call this as a growth parameter. I will call it as a growth. Okay. Now, let's take one more example. Let's take I am the example now. Okay, I am the example. Let's say last year I was 60 kgs. Last year I was 60 kg. Now from 60 kg I became 65 kg. From 60 to 65 kg my weight has increased. Now tell me in the chat quickly, all of you, is this also growth? Do I consider increase in weight as a growth, or I cannot consider as a growth? Tell me in the chat. See, first one is growth, but is this growth, students? Tell me in the chat quickly. Do I consider as a growth? Absolutely not. I cannot. Why? I cannot consider it as a growth because what if I undergo dieting? What if I stop eating food altogether? What if I start to go to gym, or if I start going swimming or cycling or running? Will there be a change in weight again? Can my weight decrease again? It can decrease again. So it is not a growth parameter, There are other examples given in your NCERT, such as a wooden log. If you take a wooden log and you keep it in water, because of the imbibition, the water will enter inside the wooden log and the wooden log will swell up because of water. Now students, is that also growth? The answer is no again. Why? Because if you take the wooden log and you keep it in sunlight, it will again become very thin. It will go back to its original state. So it is not a growth. The other example given in your NCERT students, the other example given in your NCERT is for guard cells. It is for guard cell. Guard cell students. Now, is the swelling of guard cell as considered as growth? For example, this is a typical guard cell. Now, in this guard cell, when the water enters inside the guard cell, when the water is entering inside the guard cell will completely swell up. That phenomena is called as turgid condition, turgid. Now, is that growth? The answer is no again. That is also not growth. Why? Because as soon as the water is removed from the guard cell will become flaccid. It will become back to the original state again. So what is growth, students? Growth is irreversible. It has to be irreversible. It has to be permanent change. No more going back. No more going back. So if that is the condition, can anyone give me an example here? Any example, students? But Krishna, in that case, there will not be a change in the number of cells generally, right? Tell me, students, any example? Any example for actual growth? Any example for growth, students? Let me give you the most simplest example. The most simplest example of growth is expansion of a leaf. Let's take this as a soil. Let's take it as a soil, right? In this soil, let's say I am growing a plant here. Okay. Now here I have let's take a young leaf is here. I have a young leaf here. Young leaf here. Now this young leaf will start to expand. This young leaf will start to expand. Now this is called as expansion of leaf. It is called as expansion of leaf. Now students, in this expansion of leaf, was there increase in cell size and cell number? Obviously. Now in this expand leaf, can it go back to being young leaf now? Is it possible that the old expanded leaf, can it again go back to being a young leaf, a small leaf? Is it possible to happen that? Not possible. So this is a simple example. This is a simple example of your growth. That is expansion of leaf. Okay, expansion of leaf. Expansion of leaf. Now students, one more thing is there. In this entire growth as a parameter, do you think it is metabolic pathway? Do you think there are metabolic pathways involved here? The answer is yes. Because every single growth, students, in every single growth, always remember, anabolism is always more than catabolism. Anabolic reactions are more than catabolic reaction here. Now, do you think in this entire process of growth, do you think that there is involvement of energy? Yes, there is. Because growth requires energy. We eat food to grow. So we need energy. It is energy required process. It is a energy required process. Energy required process. Now students, with that basic information, let me give you one more example. A small zygote developing into an entire adult. A small zygote growing into an entire adult is also an example of growth. Okay. Students, with all that information, with all of these information, is everyone on the same page with growth now, students? All of you understood growth? Tell me yes or no in the chat quickly. All of you understood? Okay. Now, in this entire growth, students, we need to understand and isolate which is the first step in growth of a plant. Which is the first tiny step of a growth in the plant's life cycle. Obviously, the first step in growth is the germination in the case of plants. Now, what is germination? Germination is nothing but growth of a seed. So, growth of a seed is called as Growth of a seed is called as germination. If you know, if you understand typical germination, if you see a typical germination, here we have a seed here. The seed has a cotyledon, hypocotyle. Eventually the seed will start to germinate. When the seed is germinating, you can see, eventually you can see the, here you will have the radicle will give rise to the root system and plumule will give rise to the shoot system. So the root and shoot system develops and eventually we get the entire plant. This is called as germination of a seed. Germination of a seed, the first step in the process. Now students, with that basic information, does anyone has a doubt in the basic information about growth we learned in the last 10 minutes? Anyone, any doubt, students? Does anyone have any doubt till now, students? Quickly tell me in the chat. Does anyone has any doubt till now? We read. I hope there should be no doubt because it was very basic information. Very basic information. Okay, now students. Now I'll teach you some more concepts called as growth, about plant growth. For example, plant growth is said to be unlimited or indeterminate. Plant growth is said to be unlimited or indeterminate. What is the meaning of that, students? What is the meaning of that? Meaning is very straightforward and very simple. It means that plants can keep on growing. Plants can keep on growing. There is nothing which we can do to stop the growth of a plant. Now a question to all of you, my students. Quickly tell me in the chat. What is that one parameter? What is that one feature of plant which helps in unlimited growth? What is the secret behind the unlimited growth, students? What is the secret behind it? The secret behind the unlimited growth can be divided into two categories. Okay. The secret is called as meristem. It's called as meristem. We have something called as primary meristem, as well as secondary meristem. All of you should know this from the chapter, from the anatomy chapter. Primary meristem and secondary meristem. Okay, primary and secondary meristem. Now, what is the difference here? Very simple difference. A primary meristem, students, listen to me very carefully. Primary meristem is basically present in the from origin. It is basically present from the origin of a plant. This one is later formed. The main purpose here is primary growth. Here we have secondary growth. Now you will ask me, sir, what is the meaning of primary growth and secondary growth? Listen to me. See, all of you see this diagram in NCERT. Shoot apical meristem, SAM. Root apical meristem, RAM. Now this SAM and RAM, both of these are part of primary meristem. Primary meristem is responsible for the increase in length of a plant. That there will be increase in length of a plant, that is called as RAM and SAM. SAM will include the increase in length from the top shoot. RAM will be involved in the increasing the length of the root. Okay. Now you might ask me, sir, what is this vascular cambium right here? Now student, this vascular cambium right here, it is part of secondary growth or secondary meristem. Now what does it do? Primary meristem will increase the length up and down. Secondary meristem, on the other hand, will increase the girth. Can you see here, all of you? Increase in girth, the sideways. That is the function of the secondary meristem, lateral growth or secondary growth. All of you clear on that? All of you clear on that, students? Primary meristem, secondary meristem. First point, plants, unlimited growth or indeterminate growth. Now students, second main point here, second main point. Okay, we say that plant growth is said to be open. Plant growth is said to be open. Now, what is the meaning of open, students? What is the meaning of open? Understand all. Okay, meaning of open. The meaning of open is that new cells are always added. New cells are always added. That is the meaning of plant growth is open. Second one, done. Now third one, understand all of you. Plant growth is said to be localized. Localized. Now what is the meaning of this localized? Plant growth will only happen. Plant growth will only happen in the regions where we find meristem. In other places you don't find. So in plant growth will only happen where we find meristem. Meristem. Now what about in the case of animals? In the case of animals, this will be uniform. This will be uniform. You will never find one normal child will having long neck. You'll never find. You'll never find giraffe neck in humans. Okay. So usually in the case of animals or humans, because of the genetic predisposition, the entire growth is uniform. Entire cells can grow and multiply. But unlike plant cells where we have certain cells which can grow, they are called as meristematic cells and divide. Okay. You will never find that. Is this clear, students? Is this clear? Plant growth is localized. Okay. Third one. Okay. In the case of plants, students, in the case of plants, magic happens. What is the magic? The number of organs keep increasing. Yes. The number of organs will keep on increasing. Yes, it is. Okay, I'll give you a quick example. Students, tell me in the chat, which is the photosynthetic organ in the case of plant? Tell me, which is the photosynthetic organ? If you can tell the answer, smash your like button right now. Okay. The photosynthetic organ in the case of plant is none other than the leaf. Now students, tell me, will there be an increase in leaf in the case of plants? Tell me, will there be increase in the case of increase in the leaf? Yes, there will be. Now tell me, students, which is the reproductive organ here? In the case of plants, which is the reproductive organ? That is nothing but the flower. Nothing but the flower. So flower will also keep on increasing. Plants produce a lot of flowers. Reproductive organ is also increasing. Fancy, no? Like imagine if you also do it. If heart gets damaged, new heart is formed. If brain gets damaged, new brain is formed. Liver gets damaged, liver is formed. If kidney gets damaged, new kidney is formed. Lungs, that'd be so cool. Regeneration power. Okay. Somewhat of regeneration power. Okay. Now students, understand the next point. Okay. Understand the next point. Okay. In the case of plants, oh, I think only 4 points are there. One second. I think there are only 4 points, students. Only 4 points are there. Okay. So first one is what? Unlimited growth, indeterminate. Second point is open. Third point is localized. Fourth point is basically number of organs keep on increasing. The number of organs will keep on increasing. Now next concept is called as growth is measurable. Can we measure the growth? Is it possible to measure the growth? Now students, listen to me very carefully. One technical thing I'll teach you right now. Very technical thing. Okay, listen to me very carefully, all of you. Listen to me very carefully. One technical thing I'll teach you right now. Okay. Now, when I say about growth, technically speaking, do I always mean growth in the cell size? All of you agree with me? Focus now, focus. When I say increase in growth or growth, basically, the ultimate baseline or the technical answer should be increase in the cell size and cell number. Yes. Apart from cell number, which you can easily count, can we find the increase in cell mass? For example, this is a tiny cell I have here, and this cell will eventually develop into a larger cell. That is, there is increase in the living part of the cell. There is increase in living part of cell. This living part of a cell is called as protoplasm. It is called as protoplasm. Okay. So what is happening here? From this cell to this cell, there is an increase in protoplasm. So in technical terms, I will call it protoplasmic growth. I will call it protoplasmic growth. What is actually happening here is protoplasmic growth is happening. Increase in the protoplasm. Increase in the protoplasm or the living part of a cell. Now the question here is very serious question. Can we measure this? The answer is yes, we can measure this. But there's a difficulty. Now what is the catch here? This protoplasmic growth, even though we are able to measure, but it is very difficult to measure. Very, very difficult to measure. So when something is very difficult to measure in such certain way, what do? We find alternatives. We find alternatives for measurement. And that is where these are the following parameters comes in. These are the 5 different parameters which will help us to measure the growth. For example, increase in cell size and cell number. If not protoplasmic growth, we can see the entire increase in cell size itself. One example they have given in NCERT. One small example they have given in NCERT, that is a watermelon cell. It is initially so tiny. Initially the watermelon cell is so tiny, it will increase to such a large cell. It is almost increasing 3.5 lakh times. It is increasing. We can measure this. We can easily measure this because there is a large increase. What about cell number? One example given in NCERT is the maze root apical meristem. The root apical meristem of maize, students, there is an increase in cell number, that is 17,500 cells increase per hour. We can measure that. Increase in fresh weight and dry weight. Fresh weight will always be more, obviously. Fresh will always be more because of the water content. Water content will be more. Increase in length. Increase in length can be measured in the pollen tube growth. Pollen tube growth. Increase in surface area. Simple example, all of you know. That is, for example, this is a leaf. This is a leaf right here. Okay. Now, after some time, this leaf will increase in surface area. It will increase in surface area. Yes, it is increasing. Can we measure it again? Yes, we can measure it. We can measure it now. Okay. It is possible to measure it. Increase in volume. Yes, we can measure the increase in volume. So these are the different parameters. These are the different parameters which can help in understanding or measuring the growth. Students. Measuring the growth. Okay. Measuring the growth. Now, students, give me a minute. Let me show you NCERT how much you have covered. I look at the chats, but I don't want to get distracted. Okay. See, students, we started from here. Students. Yes, we did. Okay. This is the basic introduction which they have given you. What is the growth? How is the growth? Basically, irreversible change and all. Okay. Internal, external, intrinsic, extrinsic growth we covered here. Okay. We learned about germination. Okay. See. Fundamental. It is the most fundamental. I told you. It is not defining. It is fundamental. Okay. Irreversible permanent increase. Okay. Metabolic involved. Yes. Both anabolic and catabolic both are there. But more of anabolism compared to catabolism. Okay. Energy is required. Example is what? Expansion of a leaf. I told you. When the leaf is growing. When the leaf is able to grow, there is an increase in the cell number and cell size. Example is given here. Other examples I've asked you is Piece of food is not an example of growth. Okay. Plant growth is indeterminate. What is the meaning of that? Meaning of that is plant growth will keep on happening. Plants will keep on growing. What is the secret? The secret is meristema tissue. See here. New cells are always been added. It's called as open. I told you open. This present is open is there. Okay. Shoot and root apical meristem. Done. Primary growth. Plant's primary principle for contribution, elongation of plant along with this axis, that is increase in shoot and root length. Okay. Lateral meristem is a part of secondary growth. We can measure growth. Yes, we can measure the growth. Okay. Growth at cellular level is principally a consequence of increase in the amount of protoplasm. I told you what is protoplasm? Living part of a cell. Protoplasm. Okay. Very difficult to measure directly. We have to look for other parameters. What are other parameters? Fresh and dry weight, increase in length, area, volume, cell number. Okay. A single maize root apical meristem can give rise to more than 17,500 cells in per hour. Cell size 3.5 lakhs times watermelon one. Okay. Pollen tube is for the length. So with that, we come to the end of this part. That is, growth is measurable. Now we'll be starting with something called as phase of growth. Till here, I believe there should be no doubt. So we have covered every single point from NCERT until now. Now we'll go to the next part of the chapter called as phases of growth. Can we start, students? Can we start? All of you? Can we start now? Yes, we can. Now, what are the different phases of growth? Okay. Broadly speaking, growth can be divided into three separate categories. The first one, second one, and third one. Okay. Now, which is the first one? The first one is called as meristematic phase. Meristematic phase. Second one is called as elongation phase. Elongation phase. Third one is called as maturation. Okay. Now these three are the different phases of growth in the case of plant life cycle. All three are very important. Every single phase has a characteristics which you need to learn. For example, meristematic phase. First phase. Meristematic region. It happens in the meristematic region. It does. Cells are very small here because very early young cells. Okay. Dense cytoplasm, protoplasm. Vacuoles are absent here. Why? Because vacuoles are mainly involved in storage. If there is no storage requirement, do we even need vacuoles? No, we don't need vacuoles. Okay, initially. Conspecies nucleus. Cell wall is primary here. Why? Because primary cell wall is present in the young leaf, young root, basically young cells, right? Multiple plasmodesmal connection. Now students, how many of you know what is plasmodesmal connection? Let me teach you. For example, this is one cell, this is one more cell. Within the cell, let's take here we have cell wall. Cell wall is here. We have outer cell wall. Okay. Let's just say we have outer cell wall here. And inside we have the cytoplasm. This entire thing is the cytoplasm. Entire thing is the cytoplasm. Okay. Cytoplasm. Now here students, this cell and this cell in the middle, that is called as middle lamella. Cell chapter you might have done it. Middle lamella. This cell and this cell, there is a cytoplasmic connection. That is, this right here is called as cytoplasmic connection. That is, cytoplasm of this cell and cytoplasm of this cell, both of them are connected to each other. That is, to exchange different types of substances. And this cytoplasmic connection, the other name of this cytoplasmic connection is called as plasmodesmatal connection. It is also called as what, students? Plasmodesmatal connection. All of you understood? Yes. So this plasmodesmatal connection, primary cell wall, conspicuous nucleus, vacuoles are absent, cells are small sized. All of these are features of meristematic phase. Okay. Now, second phase, where the cell is maturing a little, the cell is more advanced, the cell is elongating here. Okay. That is, the cell length increases and the size increases here. Vacuoles are present here. Secondary cell wall formation happens. All of you know how it happens, right? For example, this is a cell. Okay. This is a cell. Here we have the primary cell wall. This right here is primary cell wall. Secondary cell wall, friends. Secondary cell wall is produced on the inner side. That is, on the inner side, we always get the secondary cell wall. And this secondary cell wall will have certain type of depositions, lignin depositions, suberin depositions based on the different plant. So secondary cell wall formation is happening here. Secondary cell wall. Deposition starts. Deposition starting. Finally, the third phase is called as maturation phase. In maturation phase, every single cell has a fixed function and structure. Here, in maturation phase, every single cell has a fixed function and structure. Like they have made up their mind. They have made up their mind what is going to be my structure and what is going to be my function. Everything is set right here. Everything is set. Okay. In the region of, that is, maturation phase. Maturation phase. Okay. Now, students, you need to remember here in the maturation phase, right? Apart from differentiation, here the cell wall here will have a lot of lignin depositions. It will be here. Okay. There will be something called as change in protoplasm. Protoplasmic change will happen. What is the protoplasmic change? That is, all the vacuoles will reach maximum size. They will reach here. That is maturation phase. So there are three different phases. Every single cell undergoes meristematic phase here, plant cell, right? Plant cell. Meristematic phase, elongation phase, as well as maturation phase. So by the end of it, a cell is ready. A cell has a different function, structure, ready to take up a job. So that is called as growth phases. Now, students, if you open your NCERT, there is a concept called as proximal and distal. For example, this is the soil right here. This is the soil right here, right? Above the soil, let's say I have a stem here. Okay, stem here. Now here, students, I have a root. Let's say I have a root here. Okay, there will be a root here. Root here is there. Okay. Now this region, student, this region here is the maturation region. This one is elongation. And this growing tip is the, one second. This growing tip right here is called as meristematic region. Okay. Maturation, elongation, and meristematic region. Now listen to me very carefully, all of you. This region right here, with reference to the stem and the soil, meristematic region is always distal region. So always distal region. Region of maturation is always proximal region. Now students, I'll ask you a very simple question. Let's see how you can answer. With respect to maturation, name the distal region. With respect to maturation, which is the distal region here? So distal region is going to be elongation. Okay. Now with respect to elongation, tell me which one is the distal region again? Distal region is going to be meristematic region. Okay. One more question I'll ask you. With respect to elongation, tell me which one is the proximal region? Anyone? With respect to elongation, the proximal region is going to be maturation. Okay. Last question, I'll ask you one more. Listen to me. With respect to meristematic region, tell me the proximal region. Tell me the proximal region here, proximal to meristematic. Last question. Tell me the proximal region with respect to meristematic. Tell me. With respect to meristematic, the proximal region is going to be elongation. So this side, if you go, it's proximal. If you come below, it is distal. Distal is coming down. Proximal is going up. The basic difference everyone should know. Okay, basic difference everyone should know. Which is the proximal side, which is the distal side. They can ask you questions. Okay. See, if you look at this diagram given in NCERT, right? Here it is maturation. This is elongation, and this right here is meristematic region. Elongation, maturation. Okay. Look at this question. All of you look at this question right here on the board. It's a NEET 2023 question. Last year. Okay. Last year. Tell me in the chat quickly. Okay. Tell me. Which of the following statements is not correct? Okay. Phase of cell elongation of plant cell is characterized by increase in vacuolation, that is, vacuoles are increasing, it is increasing. Okay. Cells in the meristematic phase of growth exhibit abundance plasmodesmatic connection, they do. Plant growth is measurable, it is measurable. Okay. Plant growth is generally determinate, students. Tell me, do you think plant growth is determinate or indeterminate? Tell me in the chat. It is indeterminate. You cannot measure the plant growth. So this statement right here becomes wrong. It becomes wrong. All of you agree? So this is the easy questions which you get in examination. Easy questions you'll get in examination. So wrong statement. Option number 3. Okay. The students, I believe till here there should be no doubt till here, because the next concept which I'm going to teach you is very important, and I want 100% attention for the next 10 minutes. Can we go, students? Can we go? Next 10 minutes, I want ultimate attention now. Next 10 minutes, I want ultimate attention. Can we go? All of you drink some water. All of you drink some water now. All of you drink some water. Water break. All of you drink some water and water break. Tell me the doubt quickly. No, I'm looking at the chat. Okay. Students, like this video, quickly like the video. Structural and physiological differences with respect to what? Structural means mainly the physical things. Physiological means the hormonal reactions. Now, for example, when I teach you something called as, in the end will come, we will understand what are the different structural factors influencing the growth. When I say structural factors, I mean the photosynthetic ability, the sunlight, the water, understanding the water, the different types of parameters are there. That is structural and physical. Now there is something called as physiological. Under physiological, there is intracellular and intercellular. Do not worry, we'll come to that. What is intracellular physiological roles? What is intercellular? That is basically PGR and genetics. So if you do not worry, I will teach you today that also while we come to that topic. Okay? Nice. Now, okay. Next is called as growth rate. Very simple, students. Very simple. I need one volunteer now. Or I need one volunteer. No, not for this. Next topic I need a volunteer. This one I don't need actually. Okay. What is growth rate? What is growth? That is, growth rate is nothing but increase in growth. That is increase in growth per unit time. Per unit time, there is an increase in growth here. Okay, increase in growth. Okay, now listen to me. Okay, now listen to me, students. Quickly, all of you listen to me very carefully now. Okay, in growth rate, there are two types here. Two types of growth rate. One is called as arithmetic. One is called as arithmetic or arithmetic. Second one is called as geometric. Now, what is the difference? Listen to me very, very carefully, all of you. Very carefully listen to me. For example, this is a meristematic cell. This one right here is a meristematic cell. Now, this meristematic cells, when it divides into two cells, what happens? When this meristematic cell divides into two cells under arithmetic growth, one cell is going to be meristematic, the other cell is going to become permanent. So this right here is a permanent cell. Now permanent cell is a cell. Permanent cell is a cell which cannot divide. Which cannot divide at all. Cannot divide. Now for example, students, now this cell cannot divide, it will remain as such. This is the meristematic cell again. Now this will divide in two cells again. Divide in two cells. Now when in this division again, one cell is meristematic, other cell is going to become permanent cell. Similarly again, if this cell divides into two cells, one cell is going to become meristematic, the other cell is going to become permanent. So do you see a pattern here, students? Do you see a pattern here? All of you see a pattern? When one meristematic cell divides, one is meristematic, permanent. Meristematic, permanent. Such type of growth can be seen in the root, right? Roots. And it is a much more a constant growth, and such a growth, students, is called as arithmetic growth. All of you understood? Yes, we understood. Okay. Now, students, listen to me. What about geometric growth? Listen to me. This is arithmetic. This is geometric. Now, understand geometric. Geometric is fascinating actually. In the case of geometric, let's say this is one cell. This will divide in two cells. In this case, both the cells are going to be meristematic. Again, this divides into two cells. Further division will happen. Both the cells are going to be meristematic. Do you see that, students? Do you see that? Both the cells are going to be meristematic. That is what is happening in the case of geometric. And such a geometric growth, students, is observed in most of the plant cells and almost all the higher plants. That is the geometric growth. Now, students, what kind of question they will ask you? They can ask you the question based on the concept here, or they can ask you the question based on the graph. Can you see here? Arithmetic growth is much more constant. Can you see? Constant it is. There is no variation much here. very constant growth here. But if you look at the geometric growth, do you see here there is a lag phase, then there is a log phase, that is basically exponential phase, and then there is a stationary phase. This right here is geometric growth, much more a sigmoid curve or S curve. Sigmoid or S curve is here, this here. And if you see the formula here, in arithmetic growth, it says LT equals L0 plus RT. What is LT? Length at time t. I can call this length at time t as final length. LT equals L0 plus RT. What is L0? Let's call it as initial length or starting length. So you basically measure the final length. How do we measure the final length? We can measure the final length, take the initial length plus RT, that is growth rate or elongation per unit time, and you add both of these. When you add both of these, you will get the final length, arithmetic growth. In the case of geometric growth, LT equals L0 e to the power of RT. Here, the only thing they'll ask you, they will never ask you numerical. Let me make it clear right now. They will not ask you numerical, but rather they will ask you the formula, or they will ask you the meaning of every single terminology. For example, they can ask you, what is this E? What is E here? Base of natural logarithm. Or they can ask you, what is R? R is the growth rate. So do you realize such questions can come? And fortunately, never a question has come like in such a way that you will not be able to answer. Questions are going to be very easy, direct questions from this chapter. Okay. So I want everyone to remember these two formulas for arithmetic and geometric growth. So all you need to do is remember the formula. You're good to go. No more numerical from here. Okay. Now, this is given you in NCERT. Can you see arithmetic? One is meristematic, one is permanent. Geometric. Now here students, this is a special case which generally no one discusses. This right here is a special case for zygote. Now what is so special about it? Understand. In the case of zygote, let's say this is a zygote right here. This zygote will develop into an embryo. An embryo will develop into an adult. Now the special thing here is to notice here is that, which generally no one tells us, this zygote to embryo, the growth here is geometric. From embryo to adult, here the growth is arithmetic. So that much you need to remember. Here zygote to embryo, geometric. From here to here it is arithmetic. So please remember that small, what do I say? Question which can arise from here. Small question can come from here. Okay. Now students, see this question. Okay. I already told you arithmetic growth, students. Arithmetic growth is, example is root elongation at constant rate. Okay. Root elongation at constant rate. Here it is mainly found, it's normal geometric growth. It is found in almost all type of cells. We can see this type of growth. Okay. In almost all type of cell, this growth. See, if they ask you a question like this, right? The type of growth curve in plants. In normal plants, which type of growth curve would you notice? Okay. In all typical growth curve, if they ask you typical growth curve, your answer should always be the sigmoid or the geometric. Okay. This linear is this one. This one is linear here. This is linear. And linear is the example for root elongation. Very specific example they have given you. Specific example for root elongation. That is arithmetic. In general terms, this growth happens. That is geometric. Okay, geometric. Okay. Now students, how many of you remember when we were talking about growth measurement, we exclusively spoke about increase in surface area. Now this increase in surface area can be again broadly classified. Increase in surface area can again be broadly classified into two separate categories actually. Now I need a volunteer who can tell their pocket money. Tell me, raise your hands in the chat. Tell me who is the volunteer now. Here in ways to measure growth, there are two separate categories. Okay, the first one is called as absolute growth. The other one is called as relative growth. Okay, absolute and relative growth. Okay, let's take Varu. Let's take Varu today. Let's take Varu. Okay, now let's take Varu. Okay, before we go to that, let me tell you what exactly is the meaning of the absolute growth. Absolute growth is the actual growth which we can easily determine basically. Here, let's say I told you before. Okay, what was my, let's say my weight was 65 last year. Last year my weight was 60. Let's say last year my weight was 65. This is last year. Okay, this year my weight is 65. This year. Now students, tell me, within the span of 1 year, right? Tell me how much of weight I've increased. Tell me how much of weight I've increased in 1 year. In 1 year, how much I've increased? All you will do is final minus initial. 65 minus 60 is going to be 5 kg increase. 5 kg increase. You can tell very easily, Ninja. Yes or no? 5 kg increase you can tell me. So this is absolute growth. Very easy to tell. Very easy to tell. Okay. Now what is relative growth? In relative growth, first of all, let me tell you, this relative growth, students, this relative growth will tell you increase per unit time, and here in the relative growth, you will see the actual, not just actual, you will see the real growth here, real growth. Let me give you a quick example. Let's take Ninja, right? Ninja. Let's take Ninja's pocket money. How much you want to give it? Let's say Ninja's pocket money is 5 rupees. 5 rupees is the pocket money. Okay. 5 rupees pocket money is there. Okay. 5 rupees pocket money is there. Okay. Ninja's. Now he's like, Mummy, Papa, 5 rupees is not enough. 5 rupees I cannot even buy dairy milk now, let alone buy dairy milk for someone else. So please increase my pocket money. He'll go tell, please increase my pocket money. So parents are like, true, world has become expensive, inflation and all. So they increase the pocket money. They increase the pocket money to, let's say, students, 10 rupees. Okay. The pocket money has increased to 10 rupees. Okay. Now let's take one more example. Let's take, students, I can't even see. Let's take Christina. Let's say Christina for example. Christina's pocket money, let's say is around 50 rupees. 50 rupees is the pocket money of Christina. 50 rupees is the pocket money. Okay. Now, here pocket money increased to 10 rupees. Here also, let's say the pocket money increased to 55 rupees. Very simple. Pocket money is increased to 55 rupees. Now, students, quickly tell me in the chat, in both of these cases, how much of amount of money has increased, or is it the same? Is there any difference or is it the same? Tell me in the chat. The amount of money which is increased, is it the same or different? Tell me. In both the cases, tell me all of you. Use your mathematics skills now. Tell me. In both of these cases, 5 rupees to 10 rupees, 50 rupees to 55 rupees. Tell me, is there an increase of 5 rupees in both the cases? It is. In both of these cases, if you do 55 minus 50 or 10 minus 5, you will get as 5 rupees only. But, in both of these cases, let's take Ninja and Christina. Tell me who is more happy. Tell me, who is more happy? Even though there is an increase in 5 rupees in both of these cases, who is more happy? Question, no? Question. Who is more happy here? The answer will come from relative growth. Now, what is relative growth? Relative growth is always measured in percentage. All of you focus very well now. It is always measured in percentage. Let me show you how. Okay. The formula here is final minus initial divided by initial. Okay. Final minus initial divided by initial into 100. Now this is the formula here. Okay. Now understand. Let's do the formula apply here. What is 10 minus 5? Tell me. 10 minus 5 is 5. 5 divided by initial is how much? 5. 5 divided by 5 into 100. Okay. Tell me what is the answer. 5 divided by 5 is 1. 1 into 100 is 100. Yes or no? So here there is 100% increase. Here there is 100% increase in the money. 100% increase. 5, see, final is 10, initial is 5. So 5 by 5 is 1. Into 100 is 100% increase is there. 100% increase. Okay. Now let's do for this one. Okay. Let's do for Christina. Let's say Christina, how much? 55 minus 50 is how much? 5. 5 divided by initial is how much? 50. 5 divided by 50 into 100. That is roughly around 10% increase. 10% increase. All of you agree? So when you see on a broader spectrum, it seems like both of them, the salary is increasing by 5 rupees. 5 rupees. But what is the relative? What is the actual truth? The actual truth lies in the relative growth. One person, let's say Ninja, is getting 100% increase in the salary, 100% increase in the pocket money, but the Christina is just getting 10%. Now tell me, who is more happy now? Tell me who is more happy? Is it Ninja or Christina? Tell me who is more happy here? Students, quickly smash that like button if you're understanding these concepts. This is the most easiest way to understand this concept. The most easiest way to understand the concept of absolute and relative. Absolute will always be broader spectrum. It will never show the actual reality. A relative will show the reality of the actual increase. And in the entire, when we talk about plant growth, when I say increase in surface area of a leaf, we always use relative growth. Let me show you. This is the example given your NCERT. Here we have leaf A and leaf B. All of you can see that? Leaf A and leaf B. Just like the previous example, 5 centimeter to 10 centimeter, 50 centimeter to 55 centimeter. If you talk about absolute growth, both are same. 10 minus 5 is how much? 5. 55 minus 50 is 5. So just like in this leaf case, if you look at the absolute, it is going to be 5 centimeter increase. All of you agree? Just 5 centimeter increase here, absolute. But if you take the same, see, I have given the same value, so easier to tell. 5 to 10, 50 to 55. 5 to 10, 50 to 55. Now tell me, students, which leaf? Is it leaf A or leaf B? Tell me which leaf will show higher relative growth. Tell me, leaf will see, can you see here? Leaf A will show more of relative growth. That is 100% growth can be seen here. While here only 10% growth can be seen here, only this much. So I believe all of you understand. You cannot always take the absolute. We need to see the relative growth. We need to see the relative growth for the actual reality. Okay. Tell me, which leaf is growing more now? Leaf A is growing more. 100% increase in leaf A. By only 10% increase in leaf B. Yappa. My throat only became my throat also needs to now upgrade. All of you understood the concept? Tell me. If you understood, smash the like button first of all. Okay. Now students, now we come to something called as conditions for growth. There are multiple conditions. For example, temperature, nutrients, light, water and oxygen. All of these factors play important role in the growth of a plant, obviously. Okay, obviously students, obviously. Okay. Now students, tell me, how many of you find it difficult? Tell me honestly, do not lie to me at all. No point in lying. How many of you find it difficult to understand the concept of dedifferentiation, redifferentiation, as well as differentiation? Differentiation, dedifferentiation, redifferentiation. How many of you find it difficult to understand this concept? Raise your hands. Be honest. Do not lie. Exam is coming near. Every single concept has to be learned in detail. No more shortcuts. Tell me, how many find it difficult to understand this? Because I'm going to make it easy for you today. I initially thought I'll skip this because I've already thought this concept in plant anatomy, but then I realized there are many students who don't really understand still this concept. So let me make it easy for you. Okay, let me make it easy for you. Can we start? Small cut has happened. I don't know how I got a cut on my hand. Can we start, students? Can we start? Okay. Now. Okay. Now here I'll write and teach you. Then I'll tell you a small story to remember this. First I'll teach you, I'll write and teach you. Then I will give you a small story which will help you to remember this better. Okay. Let's start, students. Let's start. Very easy it is, very easy. First we'll start with differentiation. Differentiation. Okay. Let me use a different. I hope it is visible to all of you. I want to accommodate everything in one board. Okay, let's start with differentiation. Now, what exactly happens in differentiation? Let's say we have a meristematic cell. Let's say we have a meristematic cell right here. Now, this meristematic cell can divide. It can divide nicely. Dividing cell it is. Dividing cell. Meristematic cell. Okay, this cell can divide. Basically, cell can divide. Now, this meristematic cell, students, this meristematic cell will undergo a loss in life. Okay, it will lose the ability to divide. Ability to divide completely. Lose the ability to divide. And this meristematic cell, once it loses the ability to divide, it becomes something called as differentiated cell. This differentiated cell can also be called as, it can also be called as primary permanent cell. Primary permanent cell. And this right here is called as, differentiation. That's all. Tell me, was it difficult to understand differentiation? In differentiation, meristematic cell will lose the ability to divide. It will lose the ability to divide and it becomes permanent. That is all the concept of meristematic cell. Now, there are a few examples of this primary permanent tissue. The example of this primary permanent tissue, students, examples, understand. First example, I'll tell you, primary xylem, primary phloem, right? Primary xylem, primary phloem. Okay, then there is something called as primary medullary race. Even we also have the parenchyma, collenchyma, sclerenchyma, primary permanent tissues. They have lost the ability to divide and they are basically now differentiated cell. Okay. Now second one you understand, now second concept. That is called as dedifferentiation. Dedifferentiation. Let's take the primary permanent cell now. Let's take this. This is a right here is a primary permanent cell. This is a primary permanent cell. Now this primary permanent cell, students, this primary permanent cell, it will undergoes a rebirth. It will undergo a rebirth now. Okay, that is, it will regain the ability to divide. It will completely regain the ability to divide. Regain the ability to divide. Now, if this primary permanent cell, if it is regaining the ability to divide, then it forms something called as secondary meristem. All of you know secondary meristem, which is responsible for secondary growth. Secondary meristem. Now, here I'll give you a quick example. The example, very simple example here. Okay. Example here is medullary race. Medullary race will convert into vascular cambium. All of you remember vascular cambium? Vascular cambium is involved in the secondary growth. That's it. So, dedifferentiation is what? Primary permanent cell, it is regaining the ability to divide. Again, regain the ability to divide, forms secondary meristem. That's all. That's all is dedifferentiation. It is regaining the ability to divide. Now, understand the third concept. That is redifferentiation. Redifferentiation. Okay. In the case of redifferentiation, students, this secondary meristem, dividing cell. This right here is the secondary meristem. This secondary meristem will again lose its ability to divide. Lose its ability to divide completely again. Okay. And this particular cell now, once it loses the ability to divide, it forms something called as secondary tissue. Now, example of secondary tissue. The example of secondary tissue is nothing but secondary xylem, secondary phloem. Secondary phloem. That's all is redifferentiation. So I'll explain again. I'll give you a small story to remember this. A small story is that there is a class 12th student who cannot study further because of certain financial condition in the house. He will stop studying and find a job. So this is a class 12th student, meristem tissue, lose the ability to divide, that is, he will lose the ability to study further and he will become differentiated, and it's called as he'll get a permanent job. Why? Because of financial conditions. One boy is there. Okay, there is a boy there who is not able to study further, lose the ability to study, becomes permanent. Now this boy again, after 2 years or 3 years, once the financial condition in the house is very stable now what is he doing? He wants to study more. That is, he is regaining the ability to divide. He is regaining the ability to study again. Now again, then he becomes, let's say, he writes NEET. Let's say after 2 years or 3 years after class 12th, he writes NEET examination and he becomes a doctor. He becomes a doctor here, secondary medicine. So regain the ability to divide, he becomes a doctor now. Now after 10 years, this boy is like, I don't want to be a doctor anymore. I want to become a musician. I want to go singer. I want to go dance. Okay, change the profession only. So again, lose that ability to divide and becomes some other job he'll get, some other job. That is secondary tissue. Secondary tissue. Okay, so that is the entire thing of differentiation, dedifferentiation, redifferentiation. I'm going to move aside. I want everyone to read it once, and if you have any doubt, quickly ask me. It's very simple. It's very simple. All of you understood? Tell me, isn't it simple? So simple it is. Meristematic cell, lose the ability to divide, differentiated cell. That is called as differentiation. Primary pigment tissue, regain the ability to divide, and then meristematic tissue again, secondary tissue. Okay. Now let's say dedifferentiation. Secondary meristem, again it's gained here, it is losing here again. Lose its ability to divide, secondary tissue. So that is called as differentiation, dedifferentiation, redifferentiation. Now students, let me show you the examples how they'll ask you here. What are the examples they can ask you? Probably. Okay, see here. See. We learned growth rate, students, we learned the growth rate. We learned the growth rate. We learned about arithmetic, geometric. Zygote I told you. Okay. We learned about this formula, we learned here. Okay. Example here. Example, can all of you see this? Arithmetic growth, the following mitotic cell divides, only daughter cell continues to divide while the other differentiates and matures. So one is becoming permanent, one is dividing. Okay. Example is given here, see here. Is exemplified by root elongation at constant rate. So root elongation. Okay. A sigmoid curve is character of living organisms growing in natural environment. Alright. It's typical for all cells, tissues and organs of a plant. So sigmoid curve is a basic information for every single one of them. Every single one of them. Secondary tissue, I'll tell you example. I told you no. Secondary xylem, secondary phloem. This is what we learned here that time. Done. We learned about conditions of growth. There are different conditions. Students, you can read this. Very simple it is. Very simple it is. Conditions of growth. We need oxygen, plants need water, plants need sunlight. Simple basic elements. Okay. Now, can all of you see this example? Now see the example here. For differentiation. Okay, I'll mark it for all of you so you can read later on also. Okay. That is, example for differentiation is trachary element. Oh, it's not able to write. I'm not able to write. Elements. Okay. Lose their protoplasm. Now, what is the meaning here, students? What is the meaning here? Trachary elements. I told you xylem and phloem. If you remember, I told you the primary permanent tissue. The primary permanent tissue has lost its ability to divide. They are becoming permanent now. Differentiation. Example is xylem and phloem. Primary xylem and primary phloem. Trachary elements given right here. Given right here. Okay. Now, see, example of xylem is given here. Develop very strong and elastic cellulosic lignocelluloid secondary cell wall to carry water along the long distances. Okay. Tension. Now, dedifferentiation, understand. Okay. Dedifferentiation. Okay. See here. Formation of meristem. I told you, after dedifferentiation, we get secondary meristem. If you remember anatomy, under secondary meristem, we learn something called as interfascicular cambium and fascicular cambium generally. Okay, see, it's given here. Interfascicular cambium and cork cambium. These two, students, are the secondary meristem which are involved in secondary growth. See example given here for dedifferentiation. That is, fascicular cambium as well as core cambium. Okay, form from fully differentiated parenchyma cells. That is, this parenchyma cell is a primary tissue. Parenchyma cell is a primary permanent tissue. This primary permanent tissue will regain the ability to divide. It is regaining the ability. After regaining, this parenchyma will get converted to interfascicular and core cambium. This is given here. See, read the lines. Read the lines. For example, formation of meristem, interfascicular, core cambium from. See, it says from fully differentiated parenchyma cell. So basically from a permanent tissue fully differentiated, we get secondary meristem, that is regaining the ability to divide. So example is given here also for dedifferentiation. Okay, now what is redifferentiation? Examples. Okay. Now, the redifferentiation examples. The example for redifferentiation is the tissue culture. It is the tissue culture. So, tissue culture is a result of redifferentiation. Okay, redifferentiation. Clear, students? So, these are different examples given here. If you read just once, you will understand the entire thing. Okay, that's all. Clear? So with that, now we come to development. Okay. So please remember the examples. See, the questions will come like this. Formation of interfascicular cambium from fully developed parenchyma cell is an example of. Tell me the answer. Students, tell me the answer for this one. See, I told you, here we will get direct question. This here is a NEET 2024 question. Last year question, do you realize? Last year question, which many students could not answer. Only 70% could answer this question. Remaining 30% did not answer this question because they did not learn the example like this, which I showed you. Okay, tell me the answer now. What is the answer? Formation of interfascicular cambium from fully developed parenchyma. Dedifferentiation, we just now learned. Dedifferentiation. Tell me, are you getting an idea which type of question they can ask you? All of you? See this question, all of you. See this question now. See this question, read this question. The living differentiated cell, okay, it's a living cell is there. There's a living cell. Lost capacity to divide anymore. It has lost the capacity to divide anymore. Can regain, now it is regain the ability to divide. Regain the capacity to divide under certain conditions. The phenomenon is called as. So that is called as dedifferentiation. In dedifferentiation, they will regain the ability to divide. Right? In dedifferentiation, right? It is regaining the ability to divide. Dedifferentiation, regaining the ability to divide. Okay. So I believe till here it was very clear, it was very easy. You have some idea how the questions are going to be asked. Okay. So we can chill now. Okay. Because let me tell you, if you have watched the video till now, if you watched the lecture till now, 50% chapter is over. 50% chapter is over right on the spot. Now we are left with 50% of chapter, which I'll finish in the next 40 minutes. Next 40 minutes, the entire chapter is going to be over, and that is my guarantee. Next 40 minutes, the entire chapter is going to be over. Okay. So students, quickly tell me in the chat, are you ready for the next phase now? Where I'll be teaching you about something called as development. I'll be teaching you something called as the plant growth regulators with the help of the most amazing and convenient notes you can find anywhere. Can we start now, students? Can we start? Okay. Now I want you to download these notes very specifically. Okay. I want you to download these notes, make use of these notes. Okay. And study really well because you hardly have few days left. You cannot afford to be lazy around anymore. Okay. Now students, tell me, how is your confidence on plant physiology going on right now? Tell me your confidence level. Out of 5, tell me what is your confidence level for plant physiology at this point right now. Tell me your confidence level. Yes, bot army assemble. Tell me the bot army. All of you tell me. Students, let me tell you, I'm going to make sure you study in such a way that you do not even want to miss out any question in botany. I will tell you exactly where the question will come from and how it is going to be framed also when you do the more and more question solving with me. Okay, so if you're new to the channel, subscribe to the channel because you're in the right spot with the right teachers, right? With teachers who are passionate about their subject, who know about their subject not only at superficial level, because Gopika ma'am and I, both of us have done master's degree in respective subjects. I have done botany for more than 7 years of my life. Botany. 7 years of my life I have spent in studying botany. Okay, so you're in the right spot. Like the video, support the channel, and you will see the best results. Okay. Now let's start, students. Let's start the next part of the chapter that is called as development. What is development? Very simple definition. I'll tell you. Understand the definition. Okay. Development is nothing but all changes that occur in plants life cycle. That is called as development. Okay. Plants life cycle. For example, let's say here I have a seed. For example, I have a seed here. Now this seed will germinate. And we can see the cotyledons coming out. We can see the plumule and radicle. Then eventually it will reach a juvenile phase. Okay. Juvenile phase. This juvenile phase is called as vegetative phase. Juvenile phase is called as vegetative phase. This vegetative phase will mature eventually. It will mature. Then it will reach something called as flowering stage, where it will flower. Flowering stage. Okay. Eventually the flowering stage will end and it will undergo senescence. And eventually the plant will die. This right here is the entire plant life cycle, flowering plant life cycle. Now this is called as development. It's called as development, students, right here. Now you will understand here as it is given your NCERT also. Meristematic cells, growth, differentiation, expansion, elongation, maturation, mature cell eventually, senescence and death. This is called as development. All the changes happening in the plant's life cycle. Now students, there is a concept called as plasticity. Now what is plasticity? Now all of you understood that there is a typical plant life cycle. Now what if there is a variation? What if there is a change in the plant's life cycle? That change, right, the change in the plant life cycle or deviation from a normal life cycle. For example, every single day you wake up in the morning, you study, you eat food, you do this, you do that, and you sleep off. But suddenly one day, some random person comes and he tells you that, let's go on aeroplane, let's go to Paris, let's go to Japan, let's go to US, let's go everywhere in the world and roam around, I'll drop you back in the evening. Now, was that a change in your life, normal daily routine? Yes, there was. The change in the daily routine or a change or a deviation from a normal life cycle, that is called as plasticity. Okay, see this. Ability of plant to alter their development. Plant is altering the developmental pattern. This was a normal developmental pattern. Now, what if there is a change in this? The change is called as plasticity, students. Okay. So this change mainly happens because of two main reasons. One is environment, obviously, basically for adaptation purposes. for adaptation purposes, a plant can undergo changes in their life cycle. It can go. Okay, let me give a quick example for that. Let me give a quick example for this one, students. Now, for example, let's say this is the leaf in the aquatic plant. Okay, leaf. The same plant, okay, same plant which grows in terrestrial, environment. This is the leaf. Now, if you can see here, can you see here, students? The leaf in the aquatic and terrestrial, that is the land, there is a change in the leaf. There is a change in the leaf. Why is there a change? Because imagine in aquatic condition, obviously there will be water flowing. Water will be flowing here. Will be flowing. So this becomes very easy for adaptation. Very easy. Imagine this goes in water. It will entire thing will go back. It will go back. So this is a small change in the life cycle of the plant called as plasticity. Plasticity. Okay. Now this is found. Example for this such a plant is called as buttercup. Not the powerful girls, normal buttercup. Buttercup. Blue color one. Okay. Now buttercup is an example. Now other example is different phase of life. For example, depending upon the phase of the plant. In adult phase, there is a type of leaf. In old phase, there is a different type of leaf. Let me show you the diagram. Let me show you the diagram for that one. It's easy. See here. This one is, see, I told you this one is water habitat, aquatic, leaf is different, terrestrial, leaf is different. Plasticity because of the environment. This is because of environment. Because of environment. Now this one is because of the phase of life. Can you see? In juvenile phase, the leaf looks like this. But as the plant is growing older, there is a change in the leaf pattern. Change the leaf pattern. That is also called as plasticity depending upon the phase of life. Phase of life. Okay. See here. Phase of life. The example for this one is coriander, cotton and larkspur. Let's write down here. Example here is cotton leaf, okay, coriander and larkspur. Larkspur is the example. Phase of life. Okay. Now what is heterophyly? All of you know hetero means different, phily means leaf. For example, if a plant has a different type of leaf, it's called a heterophyly. Heterophyly. See, heterophyly. Question they will ask you like this. See, let's see who can answer this question. This type of question they will ask you. Which of one of the following plants does not show plasticity? Does not show plasticity. Cotton shows, coriander shows, buttercup shows. Answer is obviously one. Maize does not show. Or they can ask you like this. Let me read it for you. The ability of plant to follow different pathway in response to environment leading to formation of different kind of structures is called as. It is called as plasticity. Option number 3, NEET 2022 question. Okay, plasticity. Okay, but if you want to summarize development is nothing but growth plus differentiation. Growth plus differentiation is called as development in general. Okay, in general, students. Now understand, students, this entire development which you asked before, someone asked me before, can be broadly classified into 2 categories. Okay, development because of extrinsic factors or intrinsic factor. Now what do I mean by extrinsic factor? In extrinsic factor, involvement is going to be water, oxygen, temperature, then there will be nutrients. So all of these factors will play a role in development of a plant, and also light. Development of plant. Now what is intrinsic factor, friends? Intrinsic factors, there are 2 categories. One is called as intercellular and intracellular. In intracellular is mainly genetic factors. Basically size of the leaf, all the different genetic factors will be involved in intracellular. Now what about intercellular? In intercellular, main involvement is going to be PGR. Main involvement is going to be PGR. Now what is PGR? Plant growth regulators. For example, all the growth, all the development which we have learned till now, all of this growth and development can actually be controlled by a certain chemical substances. Those chemical substances also called as plant hormones. These are the chemical substances which control physiological activities. So all the physiological activities are controlled by these chemicals. Okay. For example, let's say division, differentiation, and elongation. It controls cell division, cell elongation. Certain hormones control cell elongation. Certain hormones will even control the differentiation. So all of these different functions are controlled by PGR, also called as plant hormones, plant growth regulators, or phytohormones. All of these are different names of the same meaning. Okay, they're called as PGR. Actually, let me tell you, this PGR can be broadly classified into 3 categories also. For example, there are certain PGR which can promote growth promoter. There are growth promoters, growth inhibitor, as well as both, or dual action. Let's say it's called as dual action. For example, the example for growth promoter is the first famous example is auxin. Auxin is a growth promoter. So auxin will always do what? It will promote growth. Then there is gibberellins. And then there is also cytokinins. These 3 hormones will promote growth. Okay. Now what growth inhibitor? Under growth inhibitor, we have something called as ABA, that is abscisic acid. It is called as abscisic acid, ABA. Now under dual action, a hormone which is involved in both the action, that is, it can promote and also inhibit at the same time. That is a gaseous hormone called as, can anyone tell me which is the hormone I'm talking about here, which can do dual action, which is a gaseous hormone? Anyone? It is none other than ethylene. Ethylene is the dual action. Now students, we'll be starting for the next 15-20 minutes, maximum 20 minutes, right? The entire PGR concept. Can we start now? Can we start? All of you. All of you students, quickly smash the like button. I want to see 150 likes. I have to make it 200 actually, but 150 before we finish. All of you. Can we start now, students? Last part of this chapter, where majority of the questions will come from, always, majority of the questions are always from here. Can we start now? Yes. Let me drink some water. One by one. Now students, let me tell you, I have made the perfect notes for all of you, the most perfect notes. All you need to do is download these notes and you need to remember every single concept here. If you read NCERT, there's a big confusion because in NCERT they have mentioned the entire isolation separately, function separately, and discovery separately. But I have not done that. I have included every single concept of a hormone at one shot itself. So it will be easy to study, easy to remember. Okay. Now, the first one is called as auxin. It is called as auxin. Now, auxin is a growth promoter. Auxin is a growth promoter which is derived from it is a indole structure. 1 second, students. 1 second, students. Give me a minute. One second, students. Let's start. Where's the pen? Where's the pen? Where's the pen? Got it. Now, I've written down every single main point here for all of you. So, learn these main points will be easy for you. Okay, one by one I'll tell you. For auxin, let me tell you, students, all you need to remember is the bioassay and the main functions. Okay. Now, for example, for auxin, the chemical nature is indole compounds and the precursor is tryptophan and zinc. Now, tryptophan, this particular amino acid, is the precursor for auxin preparation. Now you will understand, you will learn other phytohormones which also has a amino acid precursor. So look out for that. Look out for that. So this information you have to remember. This structure is not important. Okay. Now students, there are two types of auxins. Certain auxins are naturally produced. Certain auxins are synthesized artificially or human made. Okay. Natural auxin is right here, IAA. Okay, or IBA. What is IAA? Indole 3 acetic acid, indole 3 butyric acid. These two you need to remember. Now under synthetic, there is something called as 2,4-D. 2,4-D. Now this is the synthetic auxin which is used as a chemical herbicide to kill herbs. Basically to kill all the dicots. But it will not show effect on monocot. Remember students, this 2,4-D, it will show effect only on dicots but not on monocots. Okay, 2,4-D. Now here you need to understand the discovery. Discovery was done by Charles Darwin and Franz Darwin. Father and son duo, students. Father and son duo. Okay. Father and son duo. Okay. Now, plant they discovered in canary grass. Topic is phototropism. So they were understanding phototropism and they understood. For example, let me tell you how they did it. Right. These two scientists, Charles Darwin and Franz Darwin, students, these two scientists in canary grass, they noticed that, for example, this is a canary grass. Now here, for example, here I am adding the sunlight. Okay. Now this canary grass will grow towards the sunlight. Can you see? It will bend towards the sunlight. So they understood that there is something in the tip, something present in the tip. Because of that, it is able to bend towards the light. So what they do? They cut the tip itself. Now then they notice that without the tip, it is no more bending. Anywhere. They also did one more experiment. For example, this is the canary grass, right? They covered the entire tip. They covered the entire tip in black paper. Covered the entire tip in black color paper. Black color paper. Okay. And then also they notice. That there is no curvature. There is no curvature or there is no bending towards the light. So Charles Darwin and Francis Darwin, they finally told us that they discovered that in the shoot tip there is a chemical present, and because of this chemical the plants are able to bend towards the light. If this chemical is removed, there is no more bending. That is a discovery. But isolation was done by F.W. Wendt. Isolation of oxygen was done by F.W. Wendt. Now, how is the isolation? How is isolation done here? Understand. Isolation done. Okay. Plant, again different plant. Avena sativa, oats plant. Canary grass there. Now, what did these guys do? So, they understood that in this canary grass or Avena sativa, in this tip something is there. So, what they do? They cut this tip and they took a, let's take a beaker. In this petri dish, let's say petri dish. In the petri dish, they added this tip into this added, and they also added certain agar here, agar slab, agar. Okay. Now what happened is all the phytohormone transferred into the agar slab. It transferred into the agar slab. Okay. Transferred here. Now they took this slab they took, and they took this one stem like this. They took one more stem like this. They took one stem like this and one more stem here. And they placed this bar of agar this side here and one this side. Okay. And then there was light was falling here. And then they noticed that. If this side, if I keep the agar slab, which is all the phytohormones, this particular Avena sativa will bend the other way. If you look at this one, if this slab is this side, it will bend to the other direction. Now they understood why is it happening, why is it bending like that. Then they understood that, listen to me all of you, listen to me very carefully. For example, this is the slab right here. I am adding all the phytohormones here. Now what will happen, students, tell me? This region right here will show more growth. Tell me, yes or no? This region right here, will it show more growth? It will show more growth. Why? Because of the more phytohormones there, because of more oxygen there, it will grow. So what is technically happening here is, this part is growing faster. It is growing faster. And this region is growing very slow. So that is why there is a curvature in the opposite side. Because this region will show more rapid growth, and this will keep on growing, and this region will show very slow growth. So that is why opposite direction we can always see the phytohormones. In normal condition what happens? Is, for example, sunlight is coming from here. If the sunlight is coming from here, there will be accumulation of phytohormones in direction opposite to sunlight. If sunlight is coming from here, all the phytohormones are accumulated in the opposite side. So what will happen here? This region will grow faster, and it look like it is bending towards light. This will grow very slow. This side will grow very slow, and you can see a curvature. Towards the sunlight. Okay, because phytohormones are accumulated opposite to sunlight. Okay, remember that concept. Okay, small concept. I hope all of you remember. Okay, so that was bioassay called as Avena curvature test. It is happening. This side is there, toward this side will grow. This side is there, it will grow this side. So opposite direction always grows. Okay. Now what is the main question? The main question they will ask you is the functions. For example, root initiation, not root hair initiation, root initiation, cell enlargement, cell division, feminizing effect, provides female flowers, flower initiation mostly in the case of pineapple. There's parthenocarpy, students. Listen, this is very important. Parthenocarpy, that is, it will produce seedless fruits. Apical dominance. Now what is apical dominance? In the case of apical dominance, the plant will keep on growing in tall. Very tall it will grow. But students, tell me in the chat, what will happen if I cut the top part of a plant? Now for example, this is the plant which is growing like this. What if I keep cutting this top part? What will happen? What can possibly happen, students? If you keep cutting the top part, which mainly has the SAM, shoot apical meristem, you are cutting that part, it means that it cannot grow any tall anymore. Then it will start growing wider. It will start to grow wider and wider. Okay. Similarly, in a plant you might have seen the hedge making. In the gardens you might have seen there is a leaf wall. Okay, let me show you hedge. You might have seen this, students. You might have seen this. This is called as hedge making. This is called as hedge making. Can you see? This is called hedge. And this hedge making can be done. If you keep cutting the top part. You can make the hedge making. Okay. Prevent abscission. This is very tricky. Students. Prevent abscission. Why? Because this is little scam. This is in young leaf and in old leaf, the auxin will act very different. For example, prevention of abscission in the young leaf will prevent. It will prevent nicely. But in the old leaf, it does not. It will go on killing it. Like you go die. You're very old, time is up. Okay, that is prevention of abscission only in the young leaf, not in the old leaf. Okay, not in the old leaf. Okay, students. Okay, done. Induced flowering pineapple. Auxin can also be used in hedge making. Okay. Apical dominance. See, phenomena in which influenced by auxin and also play a major role in discovery, students. Tell me, in which of the following was used in the discovery? Used by discovery in discovery, students, of auxin. Tell me answer in the chat. Under morphology chapter it is given because roots can initiate certain types of gibberellins and cytokinins also. Okay. Students, I told you here, starting I told you, right? That is, because of discovery, Charles Darwin and Francis Darwin, they used canary grass. They used canary grass for tropism. It is growing towards the light. Okay, there are many questions like that. Let me show you. Look at this question, students. Look at this question. I think I told you already, right? Auxin is used by gardeners to prevent weed-free lawns, right? But no damage caused to. I told you, auxin mainly attacks the dicots, but not monocots, because monocots can easily metabolize it. Okay, so it does not affect the mature monocot plants. Only the dicots. Okay. A phenomena which, okay, this is done, right? Next question. Removal of apical dominance by decapacitation is utilized for. Tell me, students, what will happen if I cut the apical dominance? If I chop the apical dominance, all the plants will grow in sideways, and that is used in hedge making. It is used in hedge making. Okay. Next thing. The plant hormone used to destroy weed, that is called as, I told you, 2,4-D. 2,4-D is used for destroying the weedicide. It's our Agent Orange. Weedicide. Tell me, students, in tea leaves they do it. In the tea leaves they do it. In tea leaves they keep on removing the shoot tips. You might have seen in tea leaves they keep on removing the apical thing. They'll keep on removing it. When they keep removing the top part, lateral buds will keep on growing. Right? The effect of auxin is removed and the growth of lateral buds is enhanced. So laterally they can grow now. Laterally they can grow. Removal of shoot tip is very useful technique to boost the production of tea leaves, because it is removed, right? Okay. Auxin can be bioassayed by. What is the bioassay called as? What is the bioassay of auxin called as? Bioassay of auxin. Bioassayed by 𝐂𝐴𝐤 curvature, hydroponics, potometer, lettuce, hypocotyl elongation. Tell me, 𝐂𝐴𝐤 curvature, yes or no? 𝐂𝐴𝐤 curvature is used. 𝐂𝐴𝐤 curvature is used. It is used. Okay. Okay. So with that, students, with that, auxin is over. All you need to do is remember this following examples and you're good to go. Okay, you're good to go. Now what about gibberellins? Gibberellins. Let me tell you discovery first. Discovery very fascinating. In Japan, what happened was there's a disease called as Bakane disease. Means foolish. Baka means fool in Japanese. Okay. Now what is Bakane disease? Okay, what is Bakane disease? Understand. Okay. Now for example, imagine there is a plant, okay, there's a plant which is growing very long. There's a plant which is growing very, very long, very tall plant, okay, but it does not produce any seed. It is completely sterile. Useful or useless? Completely useless, no? Completely useless. Same thing happened in Japan. Same thing happened in Japan for a rice plant. Now this rice plant grew so tall. That it did not have any seeds, completely useless, and that is called as Bakane disease. Now later we got to know that this Bakane disease is caused because of a fungus called as Gibberella fujikori. Now this Gibberella fujikori is responsible for increase in gibberellic acid inside the plant. Okay. Now what is the chemical nature? Terpenes, indole, precursor acetyl-CoA or malonic acid. Discovery was done by Kurosawa. Isolation by Yabuta and Sumuki. Okay. Bioassay is called as alpha amylase barley endosperm test. Remember the name. You don't have to remember anything else. Just remember the name of gibberellic acid. This one. Okay. More than 100 gibberellic acid has been reported till now. GA1, GA2, GA3 until 100. Many are discovered. The first one was what, students? First one was GA3. That is first gibberellic acid to be discovered was GA3 was discovered. Okay, gibberellic acid. All are acidic in nature, formed in plastids. Now what is more important here is this part here. This part here. They will ask you question directly from the functions. If you remember these functions, you are good to go. These functions and these functions right here. These two slides are going to make you solve every single PYQ on the given till now. First one you read, first one. Stimulates stem growth. So first one is what, students? The first main point about gibberellic acid is it increases the stem elongation. Done. Second one, promotes seed germination. Third one, bolting in rosette of plants. What is rosette of plants? Cabbage, cauliflower, right? In all of these rosette plants, right, there will be bolting. For example, this is the normal cauliflower flower. There will be a bolting like this. It will start growing like this. Now basically there is an increase in the stem, bolting. Okay. Delays senescence. So it is a growth promoter. If it is delaying senescence, it is a growth promoter. Okay. Now see here. Increase in the length of the grape stalk. For example, this is the normal grape. Okay. If you add gibberellic acid, the entire stem length is increased. So there will be increasing grape stalk length. Increasing the length of the stem, thus increasing the yield. Example, sugarcane increase the yield as much as 20 tons per acre. That is, all of you eat sugarcane. Sugarcane is basically a stem. If you keep on increasing the stem length, more sugarcane, better. So it increases the internodal length here. Elongates and improves shape of apple. Speed up the malting process in the brewing industry. Spring juvenile conifers with GHGS hastens the maturity period. Students, 1, 2, 3, 4, 5, 6. All of these 6 points are 6 different previous year NEET PYQ. I am not even lying. All of these 6 points are separate PYQs. So you have to remember this at any cost. Okay. Let me give a small proof how the question is going to be asked. See this. Look at this question. Spraying of which of the following phytohormones on juvenile conifers? See, juvenile conifers help the hastening maturity period. Just now we learned gibberellic acid. The point which I told you, the same point is given here. Okay. See this question. Okay. Which of the this is a NEET 2023 question. See this question. Which of the following is not an inhibitory substance governing the seed dormancy? I told you. Which does not do seed dormancy? We know gibberellic acid promotes seed dormancy, right? Breaks seed dormancy. Gibberellic acid, directly direct answer, gibberellic acid. Okay, not an inhibitor, it is a promoter, it promotes. Yes. See this question now. Name the plant growth regulator which upon spraying on sugarcane crops, see, sugarcane crops, gibberellic acid again. So every single line there is a NEET PYQ. It is a NEET PYQ. So please learn all of these points. Please learn all of this one point. It is going to be very helpful to all of you. Okay. Delay senescence, promote seed germination, stem length, bolting rosette plants. All of these points, they're going to help you. They're going to be a best friend. Okay. Now, cytokinins. Again, chemical nature, purine derivative adenine. This right here is also PYQ. Also PYQ. Precursor is tRNA, discovery 𝐂𝐤 and his co-workers. 𝐂𝐤 and co-workers discovered it. Isolation by Miller. So one point again here. Okay. Now how did it discover? 𝐂𝐤 and his co-workers were working on tissue culture. When they were trying to grow a plant in tissue culture, it was not growing really well. But to the tissue culture, when they added something called as extract of vegetable tissue, yeast extract or coconut milk, When they added any of these following extracts into the preparation of your tissue culture media, there was a proper callus formation of tobacco, nicely formed. Then they understood that there is something is present. Something is present in all three extracts. Something is common is present. The common thing was nothing but cytokinins. So cytokinins was a common thing present in all three of them, which was responsible for cell division, responsible for the growth of callus growth. Okay, tobacco callus. So isolation by Miller. The DNA showed maximum cell division. DNA had powerful cell division promoting effect. Miller et al. Later identified and crystallized the cytokinins promoting active substance, and they called it as kinetin. And this kinetin was isolated from the herring sperm DNA. Herring sperm DNA. Okay. Now, it is also obtained from your plants also. That is, cytokinins along with auxin promote cell division. All of you know that natural substance is called zeatin. Zeatin in the case of plants. Zeatin in the case of plants. Okay. Again, functions. All you need to do is remember these functions and you're good to go. Cell regulation, done. Modifies apical dominance and promote lateral growth. Students, tell me. Cytokinins is promoting lateral growth. Tell me, is it against auxin now? Because auxin is promoting what? Auxin is promoting apical dominance, but cytokinins is promoting lateral growth. So can I tell cytokinins at some level it is antagonist to auxin? It is completely opposite to auxin in some level. It is opposite. Promoting lateral growth. Delay senescence, again, it is a growth promoter. Promote nutrient mobilization, chloroplast development, new leaves and adventitious roots formation. Okay, question will come like this. See, adenine derivative they'll ask you. So you should be able to answer cytokinins easily. Okay, cytokinins. Okay. Students, with that, third one is over now. Okay. Next one is called as ethylene. Ethylene, I'll tell you students, it is a little difficult to remember. Okay. If you read the NCERT, little difficult to remember. But I made it easy for you. How? I have made the points, the functions in a way that it affects a separate part. For example, let's get over with the first basic information. Gaseous hormone, done. Methionine is a precursor. So again, see, ethylene is also derived from an amino acid. Ethylene is also derived from an amino acid, similar to how it is derived in the case of auxin, tryptophan derivative. Same amino acid derivative. Amino acid derivative. Discovered H.S. Cousins. Bioassay is called as triple response test. Now, how did they discover? Let me tell you. For example, they took an orange here. They took an orange. They kept this orange. They kept this orange in a room with a lot of bananas. With a lot of bananas. Okay. There were a lot of bananas. So what happened? This orange right here, it released certain gaseous hormone which made the entire banana ripen very faster. That is called as gaseous hormone. Okay. That was the discovery. Effect. Students, understand. I have made effects on different paths. Effect on seed and seedling is very different from the effect on other part. For example, it breaks seed dormancy. It will break the seed dormancy. Broken. Initiate germination. So it initiates germination. Okay. Here we learned that, students. Here we learned that in the case of gibberellins, it promotes seed germination. It promotes germination, students. Promotes seed germination. Here also we are learning. Ethylene also we are learning. It initiates germination. So do not get confused which one they are asking you. Read the question carefully. If there are hints for ethylene, hints for gibberellic acid. Okay. Horizontal growth of a seedling, swelling of embryonal axis, apical hook formation in dicot seedlings. All of these are different effects on seed itself. Effect on shoot, breaks bud dormancy, sprouting of prototubers, product that comes out. Okay, this is very important students, very important. Why? Let me tell you why. Every single time you learn about internodal elongation, which hormone comes to your mind? Tell me. Every single time you learn about internodal elongation, the hormone which comes to your mind is 99% of time gibberellins. But read the statement very carefully. If the statement is telling you internodal petiole elongation in deep water rice plants, the answer should be ethylene. Do not tick on gibberellins for this. Please read the statement very carefully. Okay. Let me tell you one more hack also here. Okay. Root. Now here you need to understand, if the question is asking you root growth and root hair formation. If it says root hair formation, it is ethylene. If they tell you only root elongation, auxin. So only root elongation, auxin, root hair formation, ethylene. So please be very careful when you're reading the lines of the question. Please be very careful. One small root hair or deep water rice plants can change the answer for good. Okay. Now, what about in the case of flowering? Again, induces flowering in pineapple and mango, promotes female flower in cucumber and hence increase in the yield. Similar to auxin again, similar to auxin. Very careful. Okay, similar to auxin here also. Now, fruit, all of you know, commercially used to ripen the fruits, that is ethaphone, source of ethylene. Plant converts ethaphone to ethylene and plants use it. That is the ethylene fruit. Now, it causes senescence and abscission also. I told you, dual function. It promotes and also senescence. Okay, senescence. They can ask you a question like this. See, which hormone promotes? You will read the line like this. This is a NEET 23 question. Which hormone promotes internodal or petiole elongation? You will see this and you directly go for GA3. Yes or no? Tell me. Yes or no? You will read this line right here and you will directly go for GA3. But no. Read the line carefully. It says deep water rice plants. So answer is ethylene. The answer should be ethylene for this. So please read. See, that's why less than 70%. Why less than 70%? Because many students don't read properly. Okay. See, the gaseous plant growth regulator is used in the plants for. No. See here. Promote root growth and root hair formation. So if you read the statement very carefully, you can tell me root hair formation. Okay. Hair formation. I don't think this will be visible to all of you. Let's skip this. Okay. So that was ethylene. And type of question they can ask you in ethylene. Last topic is called as abscisic acid. One small story here. Okay. There were 3 different scientists who were working on plants and they found 3 different types of chemicals which is inhibiting the growth. The first one is called as inhibitor B, second is called as abscissin 2 and third one is called as dormin. 3 different chemicals, 3 different scientists. Later on they got to know that all of these 3 different chemicals are actually same to same. They're nothing but abscisic acid. Okay. So chemical nature is carotenoids, precursor is a bioxanthin discovery, abscissin 2, dormin and inhibitor B. 3 different scientists. Now, function. Effect of abscisic acid. Increases tolerance to stress. Also called as stress hormone. General plant growth inhibitor. It develops seed development and maturation. Inhibitor of plant metabolism. So basically growth, it is basically like poisonous to the plant. Okay. Promotes seed dormancy. It's like you wait. You don't have to germinate. I will not have to germinate. You undergo more and more dormancy. Okay. So it is completely against gibberellic acid. Completely against gibberellic acid. Stimulates closure of stomata. That is, during stressful condition, it will tell stomata to close it up. Okay. And students, with that, your chapter is over actually. With that, your chapter is over. Look at this question. They can ask a question, direct question. NEET was direct question. Which hormone is used to induce immediate closure, stomatal closure in leaves? Abscisic acid. Stomata closing. Okay. So please remember, students, please download this, all of you. Auxin, root initiation, flower initiation, parthenocarpy, everything is given here. Gibberellins is given here. Cytokinins is given here. Ethylene is given here, as well as abscisic acid is given here. The entire notes are available to you right now here. And if you want this, students, if you want this entire notes, you will get this entire notes in the description, or you will find it in my Telegram channel. Okay. So with that, the chapter is over. Chapter is very easy. You will always get targeted questions. So download the notes, study the important functions, and you're good to go. Okay. Now here is a homework question to all of you. Here on the board. So this is a homework question. 14 question. Solve the homework question. Okay. Solve the homework question, and also all the doubts, if you have any doubts, and also let me know how was the class in the comment section, and I will read the comment section and I'll clear the doubts. Or if you have anything else, let me know in the comment section and I'll be there. Okay. So thank you so much, students, for today's class, and I'll hopefully I'll see you in next class. Next class is gonna be on let's see which topic. I'm still thinking, I'm still contemplating which topic to take next, and probably it's going to be anatomy or morphology. Let's see. Okay, next class. Okay, so thank you so much, students. I'll see you next class. Bye bye, take care. Discovery of plant hormones. Discovery, they will not ask you much, right? Discovery, they will not ask you much. How much I've told you, that much they'll ask you. Nothing more than that they'll ask you. Okay, discovery is very simple. Very simple discovery. The main discovery where the question comes, no, the main discovery will only come from where you say, from the auxin. Apart from auxin discovery, right, they will not ask you anything else. Okay. Okay. If you start now, it's very difficult. Okay. Medical, I'm looking at the question. Send me the question. Medical, let me see the question. I think it went up somewhere. I can't see the question. Actually, NEET Express is going very fast. Actually, today I have a question. Today I have a class NEET Express also, right? Because the entry point is they're closing the entry points, right? They're closing the entry points for NEET Express because we have too many students there. We don't want more students more and more, because it becomes difficult to deal, because we have to distribute more, we have to do more entire thing. So I don't know how much is there. Okay, for NEET Express seats. Hopefully it is you're part of it, right? So thank you so much, students. Let me know in the comment section this answer to this question, and also let me know in the comment section how was today's class. Show some love, smash the like button, share the video, and I'll see you in the next class.